Imagine tailoring exercise and nutrition directly to your DNA to unlock peak performance, prevent disease, and increase our health span. That future begins with reading our genetic blueprint. But to do that, we first need a clean copy. Today, I'm taking you into the lab for that essential first step, showing you the complete protocol for extracting pure DNA from a key model in biomedical research, Wistar rat brain tissue. Let's begin. First, I'm taking the tissue DNA extraction kit out of the cold room. To prepare for the incubation steps, I'm preheating the dry block incubator to 58 degrees Celsius. Before starting, it's essential to be organized. I'm preparing a sheet with columns for sample ID, weight, and concentration. This data can be added to an Excel file later. I'm also making a simple discard beaker with water and dish soap for used pipette tips. Now I'm arranging a new set of microtubes in a rack and labeling each one with its corresponding sample name. With everything prepared, I'm getting my brain tissue samples from the freezer. I'm weighing out 10 to 20 milligrams of tissue on the analytical balance and placing it directly into the correctly labeled microtube. Here I'm adding 400 microliters of Ph.D. buffer to the mouse brain sample. Next, I'm homogenizing the tissue with a blue pastel until it's completely broken down. Now I'm adding the enzymes. I'm pipetting 25 microliters each of proteinase K and RNase A directly into the sample, and then giving it a quick vortex to mix. Next, I'm placing the sample into the 58-degree dry block incubator for a 2-hour incubation. During this period, I'm vortexing the sample every 10 minutes to ensure the tissue lyses completely. Alright, the 2 hours are up. I'm giving the sample a final vortex and placing it in a rack. Now I'm setting the incubator to 70 degrees for a later step. I'm adding 400 microliters of ELD buffer to the microtube and giving it a vortex. Then I'm incubating the microtube for 15 minutes at 70 degrees Celsius. This 15-minute incubation is a good time to prepare for the next steps. I'm using a special marker to label the microtubes and columns within the collection tubes, and the smaller 0.2 milliliters microtubes, which I'll use later for the nanodrop measurement. Okay, the 15-minute incubation is done. I'm vortexing the contents and then setting the incubator to 38 degrees to pre-warm our EB buffer. I'm adding 400 microliters of EBD buffer to the sample and giving it a good vortex. For this step, I'm transferring approximately 750 microliters of the microtube's contents into its corresponding column. I'm centrifuging this for 1 minute. After the spin, I'm discarding the collection tube and placing the column into a new one. Okay, now it's time for the rest of the microtube's contents. I'm transferring the remaining 700 microliters to the column and centrifuging again for 1 minute. And then, just like before, I'm discarding the collection tube and moving the column to a new one. Now for the wash steps, I'm adding 600 microliters of EFD buffer and centrifuging again for 1 minute. After that, I'm emptying the collection tube and placing the column back into the same tube. Here, I'm repeating this process with 600 microliters of the second wash buffer, ESD, and centrifuging again for 1 minute, and emptying the collection tube and placing the column back into the same tube. For the final wash, I'm using a smaller amount, 500 microliters of the ESD buffer. Again, centrifuging and placing the column back into the emptied collection tube. Now I'm centrifuging the empty column containing the sample for 2 minutes. Then I'm discarding the collection tube and placing the column into a new microtube, which I already labeled and removed the cap from earlier. To ensure the column is completely dry, I'm placing it in the hot block at 37 degrees for 5 minutes. Here I'm adding the EB buffer in 2 stages to elute the DNA. First, I'm carefully adding 15 microliters of EB buffer to the center of the column. I'm letting it incubate for 2 minutes, and then I'm centrifuging it for 2 minutes. For the second stage, I'm repeating that exact same process. This 2-step process is used to maximize the total amount of recovered DNA, as the second elution washes off any sample left behind from the first. Alright, I'm taking the column out of the centrifuge for the last time. I'm discarding the column itself, because our purified DNA is in the EB buffer that has collected in the microtube. At this point, I'm placing the cap on the microtube and it can be stored in the freezer. For quality control, I'm transferring 3 microliters into a 0.2 milliliters tube to measure the concentration on the nanodrop. We have successfully isolated pure DNA, ready for the next stage of research. But this brings up the most crucial question. How do we actually know if it's high quality? We need to measure its concentration and purity before moving on. Join me in the next video where we'll use the nanodrop and gel electrophoresis to find those answers. Make sure to subscribe so you don't miss it.
Subscribe for more.